Hey guys, Mr. Hyatt here. This is the AP Biology Chapter 43 lecture. As you can see by the title slide there, we're talking about the immune system today. So, just to sort of overview or summarize our immune system, it's basically what keeps us healthy. To keep us healthy, we're going to have to be able to recognize self and non-self. We're going to have to be able to defend against a whole bunch of things that are going to be very diverse. They're going to attack us in a whole bunch of different ways. So we have to have a really robust defense mechanism. And I think as you'll see today, we do have that. So the two major kinds of defense that we have evolved are innate and acquired immunity. Let's look at innate immunity. Anytime something is innate, that means it's something you're born with. So innate immunity, think about like your skin, for example. Your skin provides a barrier from the outside, between the outside and the inside of your body. It's not specific to one pathogen. It's not like E. coli is selectively blocked by your skin. It just provides a barrier. Those response times are going to be really quick because they're general defenses. They aren't something where you have to build an antibody or something along those lines to attack a specific specific pathogen. We also have acquired immunity. We'll talk more about this later. A lot of details about this later. But that's essentially our adaptive immunity. That's what so like you get a vaccine or you are exposed to a pathogen, and that causes you to develop memory cells, or it allows you to develop specific responses to specific pathogens. And like I said, we'll talk much more about that later. Kind of to summarize, this graphic puts it into context, I think. So our innate immunity, that's going to be things like our skin, our mucous membranes I didn't talk about, secretions, things like tears, things like sweat. They're going to, in general, wash things from our body. Maybe not wash things, that's probably not a good term, but move things from our body. And then you see our acquired immunities. Those are going to be things like antibodies and lymphocytes and things along those lines. So, like I said, we'll talk much more about those coming up. So that I think we're probably good on. We'll clean up some natural killer cells here in just a minute. This video, there's a lot of really good stuff on YouTube about the immune system. This is just one that I've used in the past that summarizes both innate and acquired immunity really well. The slide is posted on Canvas. Like I said, Google or YouTube search immune system, AP Biology, and you're going to find a ton of good stuff out there. So barrier defenses are things that provide barriers. So mucus is going to trap and allow for the removal of microbes. Think about snot in your nose. It's there to catch things so that they don't enter your lungs. Same is true of saliva to a certain extent, tears. Many of those also are hostile to microbes, whether you're talking about pH, whether you're talking about other types of molecules that we have in there, whether we're talking about excess white blood cells, things like that. Those fluids themselves are hostile to microbes. So again, those are barrier innate defenses, things that are broad spectrum. We do have these leukocytes that are white blood cells that can engulf pathogens in the body. They're going to use these TLR, which are toll-like receptors on the surface of these white blood cells. Essentially, they're recognizing self and non-self. So if we look at the picture here, that lipopolysaccharide, is going to be like our ligand. That thing binds to these TLRs. They're going to come into this. I know this looks like the nucleus. It's not the nucleus. This is going to be maybe a lysosome, something like that. So how's it work? They're going to engulf the microbe. They're going to fuse it with the lysosome to destroy the microbe. So different types of phagocytes. You see those are just specific types of phagocytic cells. Anytime you see phagocytic or phagocyte or phagocytosis, that means you're essentially eating something. You're engulfing something. Neutrophils are going to destroy microbes. Macrophages are part of the lymph system, this lymphatic system. We'll talk a lot about macrophages eventually. Eosinophils Again, more information coming. Dendritic cells, that should sound like dendrites. So they're going to stimulate development of acquired immunity. We'll talk more about that later as well. So that, sorry, went through that too quickly. Just showing the systems there, where those cells can be found. Things along those lines. So we also have these things called antimicrobial peptides and proteins. You see there are about 30 proteins that make up the complement system, which is basically what provides your innate defense, if that makes any sense at all. Interferon proteins are going to basically defend against viruses, and they're going to activate the macrophages. They're kind of like the first line of defense, is what I'm getting at. If a pathogen makes it through your barrier defenses, then these peptides have a shot at taking them down. They're going to trigger the next thing, which is going to be the inflammatory response. I've got a picture that's going to help me talk through this in a second. But notice those mast cells are important here. They're going to release histamine. You might have heard of antihistamine in relation to allergies and things like that. Histamine is going to change the blood vessels. It's going to dilate those blood vessels. It's going to bring more blood to an area, which is going to bring in more phagocytes, more antimicrobial proteins. It's basically going to open up the system so that we can work on it. On our screen here, we've got our splinter that's got bacteria on the surface of the splinter. Our skin is broken, so they've penetrated our barrier defenses. Now we've got these, where are my mast cells? There's a mast cell floating around that's releasing these chemical signals that are called histamines. Check out the size of this, probably an artery. Check out the size of it here versus the size of it here. Those histamines caused things to change, caused this to swell, caused more phagocytes to flood the area, caused fluid to flood the area, and then we get that phagocytosis where we're breaking down the bacteria, so we contain it locally. We might see swelling here. We will probably see redness here because we've got fluid and we've got blood rushing to the area. So if you've ever had a splinter, you know that it gets red, it gets bumpy, and it gets tender. So that's what we're talking about with inflammatory response. Specifically, that's a local inflammatory response. A systemic inflammatory response is a real bad deal. Fevers are an example of a systemic inflammatory response triggered by pyrogens, which are released by macrophages and toxins from the pathogens. So, like I said, we're going to get to macrophages later in the lecture. Septic shock is another overwhelming inflammatory response that's systemic. Might be one that you've heard of before. Killer cells are going to basically, they're kind of like our patrolmen. So they're going to look for anything that's not self. And they're going to destroy anything at all they come across that don't have this class 1 MHC protein. So they bump into a cell. If it has the protein, they're going to bind to it. And they're going to say, okay, you're self, you're good, so go on your way. If a cell doesn't have that protein, then they can't bind. The killer cell recognizes that they don't have this protein. You must be foreign, so I'm going to break you down and destroy you. Some pathogens can avoid the innate system. They can modify their surface. They can modify their, basically they can mimic a class 1 MHC protein. They don't have it, but they have something that's structurally close. So remember, structure controls function. Tuberculosis is an example of one of those diseases. Kills more than 1 million people a year still today. So kind of transitioning from innate to acquired immunity. We've got these white blood cells that are called lymphocytes. They are going to recognize and respond to antigens. Hopefully you know what the word antigen means. If you don't, pause the video and look it up. Or just recognize that they're foreign molecules. Lymphocytes that mature in the thymus are called T cells. Lymphocytes that mature in the bone marrow are called B cells. I forgot to go through and mark these A, B, and C. That's definitely an A-level item right there. Essentially, they're lymphocytes. They're responding to foreign molecules, and the big difference between them is where they're made. Are they in the thymus or are they in the bone the bone marrow. So these white blood cells, we're going to develop memory versions of these that are going to allow us to respond to things faster. So essentially, that's what we manipulate when we have vaccines. We get a little bit of exposure to it. Our body figures out and then remembers how to fight it. And then when we are exposed to the natural version of it, we've got our defenses already kind of beefed up. Cytokines, though, that would be definitely a C-level thing, not a huge deal. They recruit and activate lymphocytes. They're things that basically bring a lymphocyte to an area. So, acquired immunity, that adaptive immunity. Basically, we have B cells and T cells that are those white blood cells. T cells grow up in the thymus, B cells grow up in the bone marrow. They have receptor proteins that can bind to foreign molecules, and then they can break them down. This is acquired, it's specific. Each antigen is going to have a different molecule, a different type of B or T cell to break it down. Here's our definition for antigen. Hope we didn't spend a lot of time working on it. Any foreign molecule to which a lymphocyte responds. A single B or T cell has about 100,000 antigen receptors. They're identical antigen receptors. So like I said, these are specific. Looking at the structure, here's our B cell and here's our T cell. So I mentioned that the big difference is where they grow up, but they also have a structural difference as well because of where they grow up. I apologize about my images here. This is a disulfide bridge. Let me fix the text there. Okay, so we've got a B cell, notice, has this Y-shaped structure, where a T cell just has, I don't know what you call that, I guess a T-shaped structure or a parallel line, something along those lines. So you've got a C and a V. Those are going to be important. The C region is called the constant region. The variable region is what's represented by the V. So the constant region is going to be conserved across all B cells. The variable region is what makes the molecule antigen specific. So it makes our receptor the catching part, the hand part of the receptor. In terms of a T cell, same idea, but we don't have this branching receptor, so we're going to be able to receive different signals. Zoomed in version of the B cell, zoomed in version of the T cell. So all antigen receptors on a single lymphocyte recognize the same epitope or antigenic determinant on an antigen. It's essentially saying, like if I were talking about a lock and a key, I would say all locks on a single door recognize the same key. So the epitope is the key. So it's the male part of the receptor that's on the foreign invader. B cells are going to give rise to plasma cells, which are going to create proteins called antibodies that we also call immunoglobulins. So we've got our antigen that you can see is going to fit here. We've got these antigen binding sites. Oh, I'm sorry. So here's our antigen. Here's our epitopes. Those are our male versions. We've got a triangle. I don't know what you would call that one. And then a circle. A step, maybe, I don't know. But we've got these these antibodies that are going to fight the invader based upon the specificity of the receptors. Sorry, I apologize. I stumbled through that. I think that's probably a good place to leave off. I'll pick up with the next slide in part 2 of the lecture.